Hello, my name is Stephen Allen from the Department of Electrical and Computer Engineering at Brigham Young University. This talk is titled 3D Thermometry with Long Spirals and Maximized Deep Blurring. During transcranial MR-guided focused ultrasound surgery for movement disorders, surgeons use MR thermometry to observe heating, predict tissue necrosis, and protect nearby sensitive structures from being damaged. So for example, here's an image of a thermometry display used during an MR-guided focused ultrasound surgery published by Lipsum et al. The thermometry method used in the clinic possesses several advantages. It fills the entire approximately 40 millisecond time period after an RF pulse with data collection. Doing so improves the signal-to-noise ratio and precision of a temperature estimate. Additionally, this sequence uses very short readouts to prevent the heating focus from shifting due to magnetic field inhomogeneity. However, because the readout repeatedly samples the same information over and over throughout this 40 millisecond period of time, it's actually quite time inefficient, which means it inefficiently samples spatial information. There's a fundamental trade-off here. If we were to sample new information over this entire time period, we would sample much more efficiently in a temporal sense, but the susceptibility of the image to be artificially shifted or blurred increases. This forces our current clinical method, thermometry method, to only sample a single slice of information with a resolution of 1.1 and 2.2 by 3 millimeters about every 4.5 seconds. This study's purpose is to test an MR thermometry sequence that can sample a 28 by 28 by 3 centimeter volume at 1.5 by 1.5 by 1.5 millimeter isotropic resolution every 4.5 seconds. This sequence is comprised of a 40 millisecond long retraced spiral technique. The retrace technique possesses an inherent protection against shifting and blurring due to magnetic field inhomogeneities, and these effects and methods are described in the paper linked by the QR code on the screen. Our hypothesis is that this sequence, with the maximized energy deblurring technique described at the QR code here, will produce temperature estimates equally accurate as the current clinical thermometry sequence. We tested this hypothesis by placing a gel phantom at the center of an exablate neurotranscranial transducer, and then insonating this gel target with the requested acoustic power of 10 watts for 30 seconds. Both the clinical thermometry and the proposed temperature volumetric thermometry sequence were used to monitor temperature at the ablation site, at the insonation site, for a total of 6 sonications. A 6-minute cool-down time was imposed between the sonications. On to the results. For long spiral readouts, such as those used in this sequence, deblurring is critically important. These images show the estimated temperature at the point of peak measurement, peak measured temperature in the phantom, both without deblurring over here, with deblurring, and an example from the clinical Cartesian sequence. When uncorrected, the hot spot is blurred out in size and has a very low peak temperature. Deblurring using the maximized energy objective recovers the hot spot, which shows in this figure a qualitative similarity to the hot spot observed using the clinical Cartesian thermometry sequence. Here are some more quantitative results. This plot plots the temperature trajectory at the hottest observed pixel in the axioclinical Cartesian sequence in red, and then the hottest pixel observed using the spiral volumetric sequence in blue here. The temperature trajectories of the spiral sequence appear to be a little bit smoother, reflecting the higher temperature SNR and better temperature precision of a volumetric sequence, as well as some potential temporal smoothing in the temporal direction. However, the peak temperatures seem to be in the spirals sequence perhaps a little bit lower than the Cartesian sequence. Temperature trajectories in the sagittal plane are shown here in black, and the sagittal trajectories show considerable artifact and are not further examined in this presentation. If we plot the peak temperatures observed in the axial Cartesian sequence as a box and whisker plot, along with the peak temperatures observed in the spiral sequence here on the left-hand side, we can see more clearly that on average the spiral sequence possibly underestimates the lower peak temperature compared to the clinical Cartesian sequence. However, with a sample size of 6 sonications, these differences are not statistically significant even at the 0.1 level. However, here on the right-hand side, if we plot the measured full-width half maximum of the hot spot in the transverse plane using both the clinical Cartesian sequence and the tested spiral sequence, we can see that the Cartesian sequence reports a significantly narrower hot spot than the spiral sequence. In conclusion, this study found for long spiral volumetric thermometry sequences that number 1, deblurring can recover much of the lost temperature information, and that the spiral sequence tended to return wider hotspots than the clinical thermometry sequence. Future investigation can examine whether this effect is caused by residual blurring or possibly an artifact of the Cartesian scan's thicker slice thickness. Finally, I want to thank my collaborators and funding sources, including the Focused Ultrasound Foundation. Thank you.